Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the diuretic activity of given drugs to rats or mice using metabolic cage. At the end of this video, we will be able to tell the action of diuretic drugs on rats by using a metabolic cage. We will be able to tell about that the diuretic drugs removes water from the rat's body by increasing their urine output. Now, let us start with this experiment. At first, let us see the introduction part. The introduction says that the diuretics are pharmacological agents that promote the excretion of water and electrolytes from the body through increased urine output. They are commonly used in the management of conditions such as hypertension, heart failure, and edema. The above paragraph explains that diuretics are pharmacological agents, which means they're pharmacological drugs that promotes the excretion of water, which means removal of water and electrolytes from the body through increased urine output. The urine output will be increased, and they are commonly used in the management of conditions such as diseases. We are talking about diseases such as hypertension, which means BP, high BP, heart failure, and edema. Edema is the swelling of the organs, the accumulation of water in the body. Then, the present study aims to evaluate the diuretic activity of selected drugs such as furosemide, acetazolamide, osmotic diuretics like mannitol, etc., using a rodent model, rats or mice housed in metabolic cage. This means we are aiming to study about the diuretic activity. The diuretics, as we know, that the removal of water is the action of diuretic, and diuretic drugs such as furosemide, acetazolamide, and osmotic drugs such as mannitol. We are using them on rodent models such as rats or mouse, and they are stored in metabolic cages. Metabolic cages allow for the accurate collection and measurement of urine output over a specified period, enabling a reliable assessment of the diuretic potential of the test substances by comparing the urine volume and electrolyte excretion of treated groups with control groups. The efficacy of the drugs can be determined. The above paragraph says that the metabolic cages allow, what does metabolic cages do? They allow the accurate collection and measurement of urine output, which means when the rats or mouse will urinate, they are collected in the metabolic cages and then they are measured in a specified period. They are stored for in the metabolic cages for a specified period of time, and then urine output is collected from each of the rat, and then they are measured. And this is a very reliable method for assessing the diuretic activity of these drugs. And by comparing the urine volume from the and electrolyte excretion of the treated group with control group, we will divide the animals in two groups: the treated group and control group. The control group contains the rats which we will not provide any drug, and the treated groups will be of the rats on which we will administer the diuretic drugs. And then we will compare the urine output of both the groups, and the efficacy of the drugs can be determined. Now let us see the equipments required for this experiment. At first, we need our animal, rat or mice. We can use any of the rat. Then the drug. We are using furosemide of 10 milligram per kilogram. We are using this diuretic drug. And then the instrument, we are using metabolic cage. Now let us see the principle. The principle says that this experiment is based on the ability of diuretic drugs to increase the rate of urine formation by enhancing the excretion of water and electrolytes such as sodium and potassium from the kidneys. When administered to rodents, these drugs act on different segments of the nephron to inhibit sodium reabsorption, leading to increased osmotic flow of water into the urine. The above lines says that this experiment is based on diuretic drugs. We are using diuretic drug. We are using furosemide, which will increase the output of urine in urine. It will increase urine formation, and by enhancing that, excretion of water and electrolytes are increased from the body. Electrolytes can be sodium or potassium, and they will be excreted out from the kidneys. As we know, the urine production does in the kidney. The kidney produces urine. And when administered to rodents, These drugs act on different segments of the nephron, which means the drug will act on kidney. And as we know, nephron is the structural and functional unit of kidney. The kidney is made up of nephron, and these drugs acts on different nephrons to inhibit sodium reabsorption, which means sodium gets reabsorbed in the nephron, so it will inhibit the reabsorption of sodium, leading to increased osmotic flow of water into urine, which means sodium reabsorption. What happens at sodium reabsorption? The water gets reabsorbed over there. It does not get passed to the urine. It gets reabsorbed, and when we will inhibit that reabsorption, the due to the osmotic flow, it will increase an osmotic flow movement of the water into the urine. And that's how the production of the urine is increased. The animals are placed in metabolic cages, which facilitate accurate and separate collection of urine without contamination. By measuring parameters such as urine volume, pH, and electrolyte concentration over a set period, the diuretic activity of test drugs can be quantitatively evaluated by compared to standard diuretics or controlled treatments. The above line says that these animals are then placed in metabolic cages which will separately collect the urine output and without contamination they are sterilized, and by measuring these volumes on different parameters of urine, pH, and electrolyte concentration over a specific or set period of time, we can understand the diuretic activity of the test drugs as well as the control drugs, controlled animals, and we can measure it quantitatively and evaluate it, and we can compare the standards with the control and the or the standard. In the control group, as we know that we will not administer any drug, and in the standard group we will administer the drugs. Now let us see the procedure, how to do this experiment. The first procedure says that divide animals into two groups, six animals in each. We have to divide the animals in two groups, and we have to divide them six animals in each group, six in each. The first group will be of control group and the second will be of the drug group. Second step says that fast the animals for 18 hours before the experiment, allowing free access to water, which means we do not have to feed the animal anything, not even the food or water, for 18 hours before performing the experiment, which will allow free access to water. And then acclimatize animals to the metabolic cage prior to the experiment. It means that we have to acclimatize. What is acclimatize? We have to make them free around that. We have to make them feel free around the metabolic cage so that they do not panic in the metabolic cage. They will get a habitual to the metabolic cage before performing the experiment when we will put them in it for a few hours. Then administer one group with the drug furosemide of 10 milligram per kilogram to be tested, and the other with vehicle normal saline by intraperitoneal route. Okay, this statement says that we have to administer one group with the drug. As we know, we are using furosemide, so then the drug group will be administered with the furosemide drug, and the other group, in the control group, we will administer normal saline. And how we are inserting it, we will insert it through the intraperitoneal route. The intraperitoneal route is the abdominal region, the peritoneal region, and the intraparental route is inserted with the help of an injection. We insert the injection into the abdominal region so that it directly is inserted into the stomach. Then after that, place the animal in metabolic cage and then record the total volume of urine collected after 5 hours or up to 24 hours for prolonged effect. We have to collect the urine for 5 hours or up to 24 hours. And then we will compare the urine output between vehicle treated and the drug treated group. We will compare the urine output between both the groups, test and drug, control and drug. Let us see the conclusion. The diuretic activity of the tested drugs was successfully evaluated using rodents housed in metabolic cages. The study demonstrated that the test compounds increased urine output and electrolyte excretion compared to the control group, indicating their potential diuretic active effect. By comparing the results with a standard diuretic, the relative efficacy of the test drugs was determined. These findings support the use of metabolic cage models as a reliable method for screening and assessing the diuretic potential of pharmacological agents. Okay, we will discuss it further after performing the experiment. As we can see, this is the ideal observation table of the experiment, of the which the one column says about the vehicle normal saline ml, and the second is of the drug. This contains the output of the urine of the six rats. Now we will perform our experiment. As we can see, this is the RJPT Simlab simulatory laboratory, and here we will perform our experiment. So now at first, let us divide the control group and the drug group of rats. But at first, we will mark the mice of control group and drug group so that we can differentiate it. So now let's start marking it. As we can see, we are marking the mouse. We are using sorry, I'm really sorry. We are using mice over here, and then we will mark the mice on their tail so that we can know that we are using mice of the control group. We are using 6 mice, so we are numbering according to the lines on the that we are making on the tail. Then we will measure it on the weighing machine so that they are of an average weight. As we can see, this is of the number third. We are using 6 mice for the control and we are using 6 mice for the drug group. We will mark each and every mice, and after marking the control group, as we can see, we have separated the control group and the drug group. Yeah. These are the cages of both the groups. This is the weighing machine that we are using, and on the behind we can see this is a cage like structure. This is the metabolic cage over which we will place our mice after inserting the saline and the drug solution for the collection of the urine. Below the metabolic cages, as we can see over here, test tubes are placed. In the test tube, there are graduations prepared so that when we collect the urine sample, they are measured with the help of the graduation in ml. So now we will mark the mice of the drug group. Similarly, at like the control group, we will mark the mice of the drug group. Similarly, on the tail, one line says for the first mice, we will weigh it. Then again, we will put it inside of the drug group. Then we will take out another mice of the drug group and we will mark its tail with 2 lines, which says that it is of the second number. As we can see here, we're making 2 lines. And then again, we will weigh it on the weighing machine. And then again, we will place it inside the drug group. Just like this, we will mark all the drug group and drug group mices. Now we have marked all the mices inside the drug group, and now we'll apply saline to the control group. We will insert the saline solution to the control group mices. We will insert it with the help of a syringe, and we will insert it through the intraperitoneal route into the mice. We will apply the saline solution, as we can see here, to the mice. As we can see here, we have each and every syringe loaded with saline solution and the drug solution. On the left, we have the syringes with the saline solution, and on the right, we have the syringes with the furosemide drug solution. We will apply to all the mices on the control group with the saline solution. After administering the saline solution, we will apply furosemide drug solution to the drug group mices. We will take out each and every mice from the drug group, and through the intraperitoneal route with the help of the syringe, we will insert the furosemide drug of 10 milligram per kilogram to each mice. As we can see here, different syringes are used so that the reduction of the contamination is done. So now, as we can see, we have administered drug to both the animals of both the groups. Now we'll place mice of both groups to the metabolic cage. Now we will place the control group mice on the left side of the tables, metabolic cages, and the drug group mice on the right side cage. Okay, so now we'll open the lid of the cage and we will place the control group mice in the first number. The first mice, the marked one, into the first cage, and similarly the second mice to the second cage. As we can see here, we will place them very carefully so that it does not panic. And before performing the experiment, we have acclimatized the mice so that they do not panic or hinder the experiment. And just like that, we will place all the mice inside the cage. Now we will see the observation. We will observe the animals of the control and the drug group after 5 hours. We have to leave them for 5 hours so that the drug solution act on the mice. The drug will show its effect, and after that we will observe them. So now let us see the observation of both the groups. As we can see here, urine output has been collected in the test tube. As we can see here, marking has been made, and we can see here it's 3 ml, it's 4.5 ml, and it's 3.5 ml or 3.4 ml of the control group. And as well as we can see observe in the drug group. Here we have an increased amount of urine output. It's 10 ml. It's completely filled. It is this test tube is also completely filled, and this has 10 ml of urine output. As we can see by physically that the urine output of the drug drug groups of mice has shown increased amount of urine output. This is the observation table of both the groups. The vehicle of the rats, mice on which we have inserted saline solution, normal saline, has shown the output that the first mice has shown 2.8 ml of output of urine, the second has shown 3.4 ml, just like that 3.6, 2.8, 4.3, and 3.2. And the average of all the 6 mice is 3.35. And now the drug group's first mice has shown 9.6 ml urine output, and then the second has shown 8.9, and just like that 8.8, 10, 10.5, 9.8. And the average of all the 6 mice of the drug group is shown average of 9.6 ml. So as we can see, with the help of the output, that the drug group has shown increased amount of urine output, which means the furosemide has a diuretic activity and has shown the increased levels of the urine output in comparison to the normal group. So now we will close it over here. And now we have performed our experiment over here. Over this page, we can see that this is a written notes for the experiment so that we can read this before performing the experiment. As we can see here, the introduction part is written over here, the equipments required, the principal portion, the procedure part, the conclusion, and the observation table. Let us see the result of the experiment. The result says that increase in the average urine output in diuretic drug furosemide treated rat mice have been observed compared to vehicle normal saline. This means that the group of mice of the drug group has shown increased increased average urine output in comparison to the vehicle, which means that the control group of the mice, which we have inserted normal saline solution. The discussion says that the diuretic drug furosemide showed notable diuretic activity evident from increased urine output and electrolyte excretion compared to the control. This suggests a possible action on renal tubules similar to standard diuretics. The metabolic cage setup enabled accurate urine collection, ensuring reliable results. The above lines says that the diuretic drug which we have used, furosemide, has shown unnoticeable diuretic activity, which means we can compare it with the other group. And we have noted it by watching it physically. And it has shown an increased urine output and electrolyte excretion compared to the control group. And this suggests a possible action on renal tubules similar to standard diuretics. And for this experiment, the metabolic cages has helped us very efficiently for collection of the urine output and has ensured reliable results. And now, over this portion, as we can see here, download user manual. This is a button of the download user manual. Will download us a user manual of this experiment. As we can see, when we'll click it, we will open this introduction, this manual for the experiment that we will perform, so that we can read it before performing the experiment. About the introduction part of this experiment, that what are we going to perform, the equipments required, the principle of this experiment, and how this experiment will be performed, and then the steps for performing the experiment, then the conclusion part, the observation table part, and the discussion part. We can download it from over here before performing the experiment. And this manual is free for every student. so that they can at least have a manual for studying. And for those students, those who have subscription of the RJPT lab, they can perform the experiment in our software. So now, as we can see, we have completed our experiment. We will move it back over here. And now we can see that we have completed our experiment. I hope you that we have learned a lot in this experiment about the diuretic activity on mice by using a metabolic cage. And I hope that you have liked this video. And for more such learning content, subscribe to our YouTube channel, RJPT Informatics. And for more such learning content, you can follow us on our LinkedIn profile, in Instagram, as well as in Facebook. We upload our learning materials and videos over these platforms. Thank you.